Okay, we're going to get started now. Thanks everybody for coming. This is a really good turnout, much appreciated. It's a real pleasure to introduce Hung-Yuan Chu, who's presently at Van Andel. I just learned is moving to Georgetown, actually, which is very good for Georgetown, I'm sure. Hung-Yuan has done some really interesting work in a variety of different things, but all related to the basal ganglia. But today he's going to talk about an important and historically somewhat neglected topic, which is primary cortical dysfunction in Parkinsonism. And he has exploited animal models to look particularly at M1, which I think is what you're going to talk about today, M1 function and structure. I should also add that this, I guess, I don't know if you're going to talk about this, this is actually embedded in a larger collaborative project he's doing with an excellent primate group at Emory. So this is sort of a larger frame of looking at really sort of very high level systems neuroscience and Parkinsonism. So thanks very much for coming.
Thanks, Roger, for the kind of introduction. And as Roger mentioned, I'm going to move, which another adventure for me. And so this talk, I'm going to really focus on this cellular and synaptic level changes in the motor cortex circuits.
There you go.
Okay.
Try it now.
I don't have to use. I can use it.
You can use it if you want to.
Yeah. Okay, it's working. Yeah. Okay. I'm just today talk really good. Focus on very cellular and synaptic level changes in the motor cortex in mouse model of media. And when we're talking about the Parkinson, of course, we no need to introduce this topic, but I just want to have a quick overview that Parkinson, is neurodegenerative disease, degeneration of a dopamine neuron in the suprastitial nuclear area, but also degeneration of the other neuromodulators like acetylcholine, nucleocellulose, adrenergic system, and degeneration of this neuromodulator closely linked with the manifestation of a variety of symptoms in the Parkinson patients. And today I would just want to highlight that between the neurodegeneration and the manifestation of this the symptom, there is a dramatic or serious synaptic or circular adaptations. And this area has been really explored in the basal ganglia field. And such circular adaptation could occur at different levels, at different scales. And most obvious is that the structure plasticity or structure adaptation could be loss of synapse, and can be loss of axons, loss of postsynaptic spines. And this kind of structure changes also normally associated with functional changes like impaired synaptic plasticity. The best example would be stronger or more easier to induce LTP in the striatal inner pathway compared to much harder to induce LTP in the direct pathway state. And another aspect of adaptation would be the intrinsic adaptation or intrinsic plasticity, and it has been reported mainly in the striatal inner pathway, striatal RFPA and GPE or STN. After the dopamine, looks like many of these neurons, like RFPAs or the STN neurons, they lose the intrinsic stability. They're even harder to get those fine extensions. And combine this structure, synaptic and the intrinsic adaptations. Eventually, this whole circuit evolves into a highly synchronized bursting pattern of activity. This kind of synchronization could happen within a specific brain area, like in ST or in PPE, but also could across brain areas like cortex, striatum, GPEST. The whole system becomes synchronized. And at the network level, this could be reflected as the strong or robust beta band activity at the bottom. You can see that this beta band activity, we all know that highly relevant to PD pathophysiology, it's very anti-kinematic and kinetic. Again, this kind of adaptation has been better studied in basal ganglia, but what happened in thalamocortical network or in cortex, we have very little knowledge, and therefore about 5 years ago, when I started my lab, maybe I thought maybe it's a good idea to look into cortex, what happened in the cortex after loss dopamine. And I found many other projects. We started a project into the literature review, and actually we found a lot of evidence suggesting that the cortex are impaired in Parkinsonian state. And this evidence mainly come from functional imaging studies, as many of you are doing here. And today I'm just I would like to highlight one paper. This is from field study lab. And in this study, they recorded local potential from the cortex of PT patient while they performed this deep brain stimulation. And what they found is that there's robust beta amplitude and coupling with the gamma amplitude coupling with beta band activity. We call phase amplitude coupling. And more interesting, this kind of coupling is very brain subregion specific. It can only detected in the motor region, the cerebral cortex, but not in the sensory areas, suggesting that the cortical changes is very unique, very specific, not a broad change caused by, for example, by development like pathology or loss of neuromodulator. So in the animal studies, and also there's a lot of evidence suggesting cortical neuron activity dramatically altered. This is the early study by George Goldberg. And in this study, they recorded from MPTP treated Parkinsonian monkeys. And this is neuronal activity from the cerebral cortex. You can clearly appreciate this highly synchronized bursting pattern of activity. And more interestingly, compared to normal healthy state, this bursting pattern activity did not invoke a movement of body parts or like the forelimb or hands. And this I think this is one of the very early study reports the functional changes of the cerebral cortex at cellular levels. And about another 10 years later, Rob Turner and his lab really further look into this question. They put a lot of efforts to identify different subtype neurons in the cerebral cortex, and they identify the pyramidal tract neuron or the cortical striatal neuron based on their projection to the pontine nuclei or brainstem versus striatum. And they noticed that the reduced firing frequency of this neuron only occurred in the pyramidal tract neuron or PT neuron, but not in the cortical striatal neurons. And this data further suggests that change is still subtype selective. And it takes almost another 10 years. We had another paper using a different approach or technology to study local cortical circuit changes. This study published 2021 using GCAM imaging while they found the 6 PhD lesion mice. And they noticed that in the normal control animals, while the animal before the animal can start reaching tasks, there's a dramatic peak in the GCAM signal from the PT neuron, from the tract neurons. However, after loss of dopamine, this peak becomes dramatically reduced, suggesting that impaired activation of the pyramidal tract neuron after loss of dopamine. So many of these observations could be explained by this classic model of cortical base ganglia circuit interaction. And after we lost dopamine, there's strong inhibition of the thalamus by the base ganglia. Therefore, that reduces excitation from thalamus to cortex. Of course, it has reduced frequency, reduced activation of cortical neurons. However, and when we look into literature, also there's some evidence suggesting that this is true actually. Yeah. When this is a paper from Thomas Bickman, and they recorded from the thalamic neurons of MPTP treated monkeys. And you can see before and after MPT treatment, the dramatic reduction in the firing frequency in this monkey. And however, we further look into the literature, this is not always consistent. There are some descriptions between studies. Some studies further even reported that increased thalamic activation after loss dopamine, which may depend on behavior state or medication state of the animal. And of course, all these studies, a more consistent observation is that this increased bursting pattern activity in the thalamus, as shown at the bottom of right. And Actually, this first impact activity, we know that they are not they are very important for neuroplasticity, but they also contribute to neurological disorders. We were wondering how this kind of burst impact activity happen in the thalamus may impact cortical circuits. And this just like a background and introduced this in our study of cellular and synaptic dysfunction in the motor cortex. And I don't think we need really go into detail about the cortical structure, and just very briefly that cortex is not one tiny structure, big structure with different layers. And in the context of the motor cortex, we have layer 1 to layer 6, but we have very tiny or even no layer 4, very small amount layer 4 maybe. And we have different subtype neurons in the motor cortex based on their projection. We have pyramidal tract neurons or intratelencephalic neurons, IT or PT and IT neurons. Where there's a complicated, interlaminar and intralaminar excitatory network within motor cortex, as well as a very complicated inhibitory network in motor cortex. So it can be very complicated, right? How does dopamine really affect this microcircuit in the cortex? So my lab really have a very straightforward idea about cortical dysfunction. We try to apply our electrophysiology approach to study how does dopamine or how the other PD pathophysiology or pathology may affect the intrinsic properties of the cortical neuronal subtypes as well as different input, mainly in a cell subtype and input-specific manner. So in the lab, we mainly use this classic 6-hydroxydopamine model of Parkinson medium. We inject this neurotoxin into this MFB, which causes almost complete loss of dopamine in the striatum, like you can see here, compared to the control hemisphere. This mice also develop very typical Parkinsonian motor deficit. You can see the forepaw in the green circle is kind of fully open, never animal rare, but not the other lesion, in the forepaw in the red circle. So we also use retrograde approaches to label PT, pyramidal tract neurons or intratelencephalic neurons, IT neurons, sorry, to based on their long-term projections. And for small amount of study, we use some report lines to identify interneural subtype in the cortex. Of course, of the genetic was used to selectively activate thalamic input or multithalamic input to the cortical circuits. So this is the overall strategy for us to activate the cortical circuit in Parkinsonian state. So we found the postdoc fellow in the lab, and what he did, very straightforward again, he with patch clamp record the beads, retrograde beads labeled cortical neurons based on their projections. In the first study, he recorded from PT neurons, and he injected different level of current. Somatic current injection invoke action potentials. By doing this, we can evaluate the cellular excitability by constructing this frequency versus current curve. You can appreciate that dramatic reduction in cellular excitability of PT neurons from the lesion animals, stressing the intrinsic excitability of PT cell impaired in Parkinsonian state. Next, we record from the IT neurons, same strategy, but different subtype. And you can see in the green curve there's no dramatic change compared to controls, suggesting that there's no significant alteration in the IT neuron excitability compared to controls. And we further analyze the intrinsic properties of the PT and IT neurons. We notice that in the PT group, there's increased relay-based current, suggesting means you need a large amount of current to invoke a single action potential. The action potential threshold more depolarized, and action potential become much wider. And also these action potential spike trains become stronger, show stronger spike adaptation compared to these controls. You can see the regular firing compared this gradually increase into spike intervals. So we thought this is very interesting observation regarding this cell subtype selective changes in the intrinsic stability. However, there's still why this is important for PD, right? It's still a question how a single cell level changes may affect the cortical network. We have an answer, and what we do to study this So we talked to our computational neuroscience in the team, Dr. Bill Layton in Sony downstate. And in that team, they did a good job that they built a computer simulated M1 model, which about 300 micron micron in volume. And this in this model, they integrate all the detailed information from electrophysiology recordings from early recordings of about cortical PT and IT neuron, the morphology, detailed morphology information, detailed thermal size, and distribution of dendritic branches across layers, as well as the ion channels at basal dendrite versus apical dendrite. Very detailed modeling of the M1 network. They also integrate these the different cell subtypes across layers. They have more than 10,000 neurons distributed across 6 layers modeling and more than, I remember correctly, 30 million synapses in this model. So we thought this is a very powerful tool to understand how single cell activity changes here may be amplified at network level. So we talked to him and let him unregulate intrinsic excitability of layer 5 PT neuron to the level that we observe in our recordings. And let's run through the HPC for a couple of days, and then let's see what they have. So, very surprising to us that when we downregulate this PT neuron intrinsic excitability, in this model, we got a kind of slightly increase, but significant increase in vivo like activity in this model, and which not affected. We also, more interesting that we also see that a simple downregulation of the intrinsic excitability of PT neurons, you can promote this beta field activity in the simulated M1 network, which are highly relevant to PT pathophysiology. And as I just want to use this as a good model as an example to highlight that this computational model is a powerful tool for us to study how single cell synapse level changes may impact this network level dynamics. And in this model, they assume that the thalamic excitation, as indicated by the green arrows there, is not affected by loss dopamine in the Parkinson state, but we don't know. We check the literature. There's no information about this connection between the thalamus and the cortex when they built this model. So the design experiment tells how loss dopamine or Parkinsonism affect the connection strength between the thalamus and the cortex in this model. Of course, we use optogenetics to inject AAV virus to express channelopsin into ventral medial part of the thalamus. But however, you can see that the virus spread to VL as well. We call this region, the green region here, the motor thalamus, which includes both VL and VM in this study. And we got a very typical, this thalamic exon terminal distribution in the motor cortex, which concentrated in layer 1 and layer 5. And after doing this, we can record some retrograde labeled PG and IT neuron and measure the synaptic drive from thalamus to PT and RT cells. So this is data from recording from the labeled PT cells, and we measure the amplitude of postsynaptic current to assess the connection strength between thalamus and the recording neuron. And you can appreciate that the amplitude of EPSCs is from 6-OHD mice is much smaller compared to controls, and suggesting that the loss dopamine reduces the synaptic drive from thalamus to cortical PT cells. And then we record from the IT group. IT neuron were labeled by tracer injection to the contralateral striatum, and we perform patch clamp recordings, voltage clamp recording, and assess the amplitude of EPSCs. And you can appreciate that there's almost no difference between this control and the lesion eyes. Again, this data suggests that there is a cell subtype selective reduction thalamic drive of the cortical circuits. And further studies stating that such reduction not only cell subtype selective, but also input specific or synapse specific. So in this study, we express channelopsin in the sensory cortex, and we record from PT cells again, and we quantify how this loss dopamine affects sensory cortical input PT cells. And you can see, again, we use this amplitude of EPSC as a measure of thalamic drive, we've got no difference between control and the lesion animals, again suggesting that low dopamine does not affect sensory cortical input to the M1 PT neurons. We also performed this the other additional physiology study and get some idea that after low dopamine, the number of functional connectivity between telomeres and the cortical PT cells will decreased. So next we need another approach to further validate this idea, and we use immuno approach, and we use a VGLUT2, vesicular glutamate transporter 2, as a marker of thalamic axon terminals in the cortex, the cortical mainly cortical layer 5. And we also label the postsynaptic spine using a fluorescent dye and quantify how low dopamine affect pre and postsynaptic structure. And here the data. This is the green puncta, the VGLUT2 immunoreactive puncta here. These are from control, these from lesion mice. You can appreciate that the number of these puncta decreased dramatically after low dopamine, and stressing that then either the expression of VGLUT2 protein itself or the presynaptic axon terminal decreased after low dopamine. And postsynaptically, we use different approaches to label spines. In this study, we use biosighting to fill the PT neurons, so we can see their spine here. You may be able to appreciate that the controls, we have a lot of spines, but not in these lesion animals. So now we have lots of presynaptic structure, postsynaptic structure, which likely, still perhaps, there's lots of synapse between telomeres and the cortical PT neuron in Parkinsonian state. And I would like to highlight that similar study has been reported by Dr. Udall Smith at Emory University. He also did a talk last year. You may still recall. They also report this loss of VGLUT2 immunoreactivity in the deep layer of the monkey of M1. And so now we have PT neuron, right? PT neuron, we have the PT neuron, these receive input. Loss dopamine selectively reduce thalamic excitation to PT neuron, but not the sensory cortical input. We also want to know why and how this could happen. What's the mechanism, in other words? So, we're electrophysiologists, and the only thing we can do, or the best thing we can do, is we can compare their synaptic properties, whether these two synapses have different properties or not. So actually, looks like it's correct that these two synapses have these very distinct synaptic properties regarding their postsynaptic AMPA-AMD receptor compositions. So in this study, we recorded this EPSC from PT neurons at plus 40 millivolts and in the presence of antagonists for GABA receptor and AMPA receptor. So this is isolated AMD receptor mediated component. And then we apply this arfenprodiol, which is a GLUN2B AMD receptor antagonist. You can clearly tell this drug could effectively suppress AMD receptor component in the PT, in the thalamic input to PT neuron, but had little effect to the sensory cortical input to PT neuron, suggesting that at this pathway, thalamic excitation to input to PT cell, we have much more GLUN2B AMD receptor. On the other hand, when we record this anti-receptor mediated component at the -80 millivolts from PT cells, and we apply this AASPM, which is another antagonist for GLUN2 lacking calcium permeable AMPA receptor. This chemical could effectively suppress the EPSCs, AMPA EPSCs, at a sensory cortical input to PT cells, but had a little effect to the thalamic input to the PT cell. To summary, what we found through this study is that at thalamic input to PT cells, we have more GLUN2B containing AMD receptor. In contrast, we have at cortical input to the PT cells, we have more GLUN2 lacking calcium permeable AMPA receptor. So this is we were very excited by this data, mainly because we already know that GLUN2B and GLUN2B AMD receptor contribute to a variety of neurological disorders, and in my own study during the postdoc fellow, we also noticed that overactivation of AMD receptor at the cortical ST pathway could mediate loss of synapse in that pathway. So therefore, we designed a different experiment, and artificially or artificially overactivate the AMD receptor. And this pathway actually can reproduce a lot of phenotype in this particular state. So to explain why this is important in the context of PD pathophysiology, and we believe that after loss dopamine, yes, the rhythmic bursting pattern of activity in telomeres that triggered a large amount of glutamate release in cortical areas because of a high level AMD receptor at the thalamic to PT neuron pathway overactivate this AMD receptor, which triggers downstream signaling pathways, leading to this weakened thalamic input to PT cells. However, in the cortical pathway to the PT cell, we have more resilient because they have less AMD receptor. This is how our current working model is done on this part.
Do you think that's leading to sort of synaptic retraction?
Yes,
Yeah,
I think
yeah,
that is what
yeah.
we saw at this moment.
Okay.
Yeah.
Interesting.
And speaking to this synchronized or rhythmic bursting pattern of activity in telomeres, we also designed the chemogenetics experiments to block the beta-gamma output to the monotelomeres. Actually, we can rescue a lot of phenotype we observed in Parkinson state. And this work has been published last year, and if you're interested, feel free to check more details in the paper. And now, just a very quick summary about how the loss dopamine affect or remodel cortical circuit. So we see that after loss dopamine, there's reduced intrinsic stability of M1 PT neurons that reduced thalamic excitation to M1 PT neurons, but no change in the cortical excitation. And we did not see any changes in the IT group. Again, this observation is very, very important. It can explain the reduced iron frequency in Parkinson state of PT cells, but it's still kind of hard to explain how they synchronize the bursting kind of activity of PT cells and realize that with them happily. So we must miss something. And we might, and the question would be, what's the missing piece in this whole picture that may lead to this highly synchronized pattern of activity? So we go back to this our computational neuroscientist and ask him what else he noticed in a model. And they said this beta band activity actually largely depends on the interlaminar excitation, and also depends on the layer 5 PV cells. And at this moment, from my lab, it's a little bit hard to study the interlaminar excitation, but we can study PV cells at this moment. So we have another project really study how loss dopamine affect interneuron in the cortex. But in the same animal model, unilateral 6-hydroxydopamine lesion, and try to study how loss of dopamine affect in different parameters of PV cell, their number, their morphology, their excitability, their connection with pyramidal cells, and their excitation. So we're going to go through this list very quickly. And Suraj and Gabe, they form a small team, they took care of this project. They used PVT tomato mouse line to see these PV cells. And actually, this T tomato is very reliable to report PV expressing cells in the motor cortex. And the first thing they did, they count the cell numbers in the motor cortex and to see whether that loss of PV cells after loss dopamine. And this image from control, this image from the dopamine depleted mice, you can visually tell that no significant cell death after loss dopamine in the cortical PV cells. And then they label these PV cells using biosighting. They can reconstruct their morphology. And here are some examples from reconstructed PV cells. Layer 5 PV cell that we mainly focus on layer 5 here. From controls and from PD mice, and you can appreciate that visually that also no change in the overall morphology. And we quantify that dendritic length, dendritic field, and branch points, that kind of thing, are also somewhat size, and all no change. And no change in the morphology of PV positive cells. Again, we are electrophysiologists. Let's record from these cells, from these tomato labeled cells in the motor cortex. And you may think that PV cells, they are fast sparking neurons, but actually in our data set, we saw a variety of different firing patterns, but they are not always fast sparking. Some cells they fast sparking, some cells they start firing, they fire, they pause, they fire again. Actually, very hydrogen, sugar, lot of hydrogenated. So we went back to the literature and actually there was a paper published in 2018 suggesting that there's some subgroup of PV cells actually project to the contralateral hemisphere. You can see that each labeled PV exon in the corpus callosum, and actually it's roughly about 40% PV cells project to the contralateral hemisphere. It's a large proportion. So now we have two subtype PV cells in the frontal cortex. One is classic PV interneurons, the other is PV-CC, of which project to contralateral hemisphere. So we are wondering, can we distinguish them in our data set? So we use cluster analysis. We just organize our data and cluster the data based on the electrophysiology properties like exponential parameters, membrane properties, and then we use a different variety of algorithm. We can always get two clusters of cells, and this is K-mean clustering. This is unsupervised hierarchy clustering. And when we check the literature and map back to our dataset, we can see that one cluster belongs to PVCC, which project contralateral hemisphere. The other cluster belongs to the interneurons. They are very different regarding a variety of different membrane properties. The PVCC, they're bigger because they have much bigger cell capacitance. They have a lower input resistance, and they also have much higher real-base current. The PVCC cells, which project contralateral hemisphere, they also have a narrow and a lower, smaller action potential compared to classic interneurons. So the question would be whether those dopamine affect both or one or either subtype of PV cells. And we compare them, and a little surprising, we did not see any difference. We did not see any difference, a major difference in the input resistance and the real base current. You can see the control data is a depleted animal. And you can see between CCs, between the interneurons, there are no dramatic change. The only difference we see is this PVCC cells, which project contralateral hemisphere. Looks like after loss dopamine, their cell body becomes smaller. The only difference so far. Yeah. To the conclusion that loss dopamine does not affect the intrinsic properties or cellular excitability of the PV cell in the cortex. We know that the PV cell in innervated pyramidal cells is through this parasomatic innervation, which is very strong or powerful GABAergic inhibition of pyramidal cells. And in this study, when we record from these putative pyramidal cells from these channelopsin, EYFP animals, and we can depolarize the pyramidal cell by current injection and shining the blue light to activate this parasomatic inhibition. You can see that this parasomatic innervation could stop cell firing by either long period light or short period light, which can be abolished by the GABA-zinc, which is GABA receptor antagonist. So we're wondering whether those dopamine affect this parasomatic inhibition. The first thing we did is we just count the number of the parasomatic puncta. In this study, we label the cortical PT and RT neuron by retrograde virus to express EGFP in PT and RT cells, and then we will quantify this puncta. And you can see we do not see any change in the number of parasomatic puncta in PT and IT neuron after loss of dopamine. And we further perform electrophysiology recording from PT and IT neurons and activate PV exon terminals around the cell and measure their inhibitory kinetic current. Again, we did not see the change in the amplitude of GABA iPSCs from controlled dopamine depleted animal EPT cells, simply for the IT group as well. So the conclusion would be that no change in the parasomatic inhibition of cortical pyramidal cell after loss of dopamine. It may be a little disappointed so far, but the last step is thalamic excitation to PV cells In this study, we inject channel dopamine into thalamus and record from PV cells here. So we using optogenetic, we can quantify the connection streams between thalamus and PV cells. And here the data actually, we immediately can appreciate that there's dramatic increase in the thalamic excitation to PV cells after loss of dopamine. You can see such difference in the examples here as well from the summary graph over there. The visual got something exciting. So this table at the bottom summarizes what we know so far. After loss of dopamine, there's no change in the cell numbers of cortical PV cells, and no change in morphology, no change in their intrinsic membrane properties, and no change in the inhibition of pyramidal cells. But there is an increase in thalamic excitation. This is very, very interesting, in my opinion, mainly because, just very briefly, this is the normal healthy state showing the macro graph, macro circuit in the cortical circuit formed by PT cells, PV cells, and maybe some SST cells over there. We have no time to discuss that part. And this thalamic input innervates both PT and PV cells, and PV cells inhibit PT cells. And so our data so far suggesting that after loss of dopamine, there's a decrease in intrinsic excitability of PT cells that decrease thalamic excitation to PT cells. The inhibition between PV and pyramidal cells are not affected. In the excitation to PV cells, they are much stronger after loss of dopamine. And we know that PV cells, they are very powerful in gating the pyramidal neuron activity, in the physiologic state, and no change in the inhibitor connection between PV and the PT cells, suggesting that the infrastructure is still there for PV cells to regulate pyramidal cell activity. In Parkinsonian state, this abnormally strengthened excitation to PV cells may contribute to the generation of this rhythmic bursting pattern of activity in pyramidal tract neuron in Parkinsonian state. And of course, this should be further validated either using experimental approaches or using computational approaches. Okay, in the last few minutes, I would just like discuss some pilot study in the lab. So Parkinson's disease is still a progressive, chronic, progressive neurodegenerative disease. And this graph here shows the loss of T-checks of terminals in the striatum in the PD patients. It takes years. And our model cannot model this. In our model, dopamine degeneration only occurs in a week. And therefore, we want to use another model, better model, to better reflect this chronic degeneration. Yeah, this model we call it, now we call it people already generated in the field for more than 10 years already, called multi-path mice. Initially, you can see that at 6 weeks old, these mice have normal level of negral structural axon terminals compared to controls, but about 20 weeks, you can see that you can appreciate almost no dopamine axon terminal in the dorsal striatum, and this animal can live up to like 40 weeks, by which time you have to sacrifice them. And so therefore, we use this model to study cortical adaptation at different stages of Parkinsonism. This model was generated by cross T-fam flux line with that query line. And you can see by 25 weeks, there are almost no healthy dopamine neurons in the negral area in the multi-path mice. And the panel over there shows the progressive loss of striatal dopamine axon terminals in the multi-path mice. At 8 weeks, it's normal, comparable to control animals. By 14 weeks, there's about 40 to 50% reduction in the dopamine axon terminals in the striatum. And at 18 weeks and beyond, there's nearly a complete loss of dopamine axon terminal in the striatum. And this animal, of course, you can expect they show progressive decline in their locomotor activity. They have worse actual performance in root rot test compared to controls. So, as Roger mentioned earlier, this is part of this ESTEP project. We try to study how the cortical circuit adaptation occurred at different stages as progressive of dopamine. we predefined a few time points, like 6 to 8 weeks in normal healthy state, at 12 to 14 weeks, at which moment this striatal-dopamine exon terminals lost about 50%. The animals just show the detectable or noticeable motor deficits. And the last point, third one is 16 to 18 weeks. These animals show a nearly complete loss of striatal-dopamine, which is severe motor deficits. And the last time point is beyond 24 weeks. We call it end stage or last advanced stage Parkinson median. We applied in vivo calcium imaging as well as active brain slide physiology recording to this mice to try to understand the cortical circuit dysfunction in this model. And today I'm going to really focus on the ex vivo part. And from the 6-hydroxydopamine model, we know that lost dopamine dramatic decrease of the thalamic excitation to M1 PT neurons. So in this model, we just go directly to this pathway. We inject tranodopsin into the ventral medial part of the thalamus, and we record the excitatory postsynaptic current arising from the thalamus, the PT cells. And we perform this study at 8 weeks from control. And we recorded from both M1 and M2 this time. And at 8 weeks, you can appreciate that the Parkinson mice, this show almost identical thalamic excitation compared to controls in both M1 and M2. And when we repeat this experiment in the 14 weeks Parkinson mice, the same thing, no change in the thalamic excitation to M1 and M2 PT cell. Keep in mind at this time point, there's about 50% reduction in the striatal dopamine exon terminals. Then we wait for a long time, like 18 weeks, 16 to 18 weeks, we start to see some reduction in the M1 PT cells. But in the M2 PT cell, that's even stronger or robust reduction in thalamic excitation compared to M1. Looks like M2 may be affected at an earlier time point compared to M1. The last time point is 25 weeks. You can both M1 and M2 PT cell, they should significantly reduce thalamic excitation compared to controls, which is very, just mimic what we got from the 6-hydroxydopamine models. And to summarize what we have from Parkinson mice, looks like as progressive loss of striatal dopamine, there's a progressive reduction of the thalamic excitation to M1 and M2 PT cells. And maybe we still need to confirm that M2 PT cells may be affected at earlier time point compared to M1. We also studied the intrinsic excitability of PT cells, which show actually dramatic reduction in the intrinsic excitability of the PT cells, which was at 24 weeks of M1 PT cell, but like 18 weeks for M2 PT cells. So we think that this cortical circuit is also very, very important for PT therapies. So if we all actively contribute to the L-DOPA media therapeutic effect. Right. So in this study, we treated the end stage Parkinson mice, which were symptomatic, and with low dosage of L-DOPA, like 4 milligrams per kilogram for 5 days. Then we perform electrophysiology recording from these animals. And these behavior data, locomotion tests every day. You can see from saline treated animals, Parkinson mice perform very bad or worse compared to controls, which was expected. And in Parkinson mice, you can see initially on day 1 and day 2, here, the Parkinson mice also perform not as good as control. But after day 3, Parkinson mice catch up to the control level, just L-DOPA gradually restore the motor function in Parkinson mice after a period of time. So with that, after 5 days treatment, we perform electrophysiology, we measure thalamic excitation to M1 and M2 PT cells, and this data from M2 PT cells. And you can see from saline treated Parkinson mice and controls, we got smaller thalamic excitation, which was just like what we discussed before. But in the L-DOPA treated mice, you can see what L-DOPA could basically restore this impaired thalamic excitation to M2 PT cells. And similar observation was also seen in this M1 PT cell. It looks like our L-DOPA treatment which restore this impaired thalamic excitation to cortical PT cell.
Have you looked at the morphology for that?
Not yet.
Not yet.
Next step.
That would be really interesting.
Yeah. Well, we're seeing green, one of green to primary.
Good for that. Yeah, so you're on, well, that's great.
So what kind of project? So, you know, in the future, next step, we are going to try to repeat this study at early stage.
Right.
Yeah. Whether we can, if we treat the microbiome like 14, 16 weeks, delay the onset, or rescue or prevent this impaired thalamic excitation. Another major question we still have no answer. How this impaired thalamic excitation contribute to the manifestation of PD symptom? Which aspect PD symptom are closely linked with impaired circuit dysfunction? We don't know yet. So I just want to summarize the information from the mouse mice, and so far, we can basically reproduce the reduced thalamic excitation to M1 and M2 PT neurons using the progressive model of Parkinsonism. The conclusion is that would be the acute or progressive loss dopamine induces very similar cortical dysfunction after loss dopamine. Another one, maybe, regardless of disease progression, time course of disease progression, cortical may exhibit very similar circuit adaptation in Parkinsonian state. The second part is that the impaired thalamic excitation or input to cortical PT cells can be rescued by chronic L-DOPA treatment. And in other words, maybe this cortical circuit may contribute to this biological, or maybe part of the biological mechanism associated with L-DOPA therapeutic effect. And I would just like to take this opportunity just to see that loss of dopamine or Parkinsonism trigger the various subtype and intraspecific change in the cortical circuit. And we identify that AMD receptor is the key molecule, could be one of the key molecules that mediate the remodeling of cortical circuit in Parkinsonian state. I have no time to discuss, but we also show that chemogenetic block with output progressive cortical dysfunction. In the second part, we show that L-DOPA can also rescue this impaired thalamic excitation to M1 pyramidal cells. So, and with by combining the data from two different models, we believe that both acute and chronic loss of striatal dopamine could induce this very similar cortical circuit adaptation in Parkinsonian state. And with that, I would like to acknowledge the people who really work on this project, and which we highlight with the arrows here, and also show the pictures from the top. These are our big ASAP team, and from my lab, and as well as Thomas Wickerman and Eula Smith and Adriana from Emory, and Bill, our computational neuroscientist from SUNY State, and from there is our funding agency. And thanks for attention.
All right, thank you. That's great. So about the money, that's in a Black 6 background, right?
Yes.
Yeah. Okay.
Yes.
Yeah. And have you tried a longer treatment than 5 days? Or?
Not yet. Yeah. This is a very fresh data. We always
right.
treatment. Yeah.
Well, because there's Josh Dudley did that, right? But he actually treated for a couple of weeks, and actually there's a substantial sensitization towards the end of that interval, which was really quite an impressive response. And I think actually resembles some of the human pharmacology, which is not widely appreciated. So
yeah, I agree. I think in our animal, in this treatment paradigm, we also see some hypersensitivity towards the 4th stage, 5th stage. Actually, the animal. Yeah, I've actually moved much more than controls on 5th stage.
Interesting.
Yeah,
okay. I think Omar has a question. Yeah. Omar.
Yeah, great talk. Just a couple of questions actually, and I'll go one at a time and let you just answer each one. First, a technical one. When comparing the EPSC amplitudes, in 6-hydroxy treated versus control animals. How do you normalize their amplitudes? Because as you know, with optogenetic injections, the postsynaptic response can vary from animal to animal. So how do you control for that?
Yes, yeah. That is a very good question. Yeah. Right now?
I think Momar has to mute himself, so.
I am muted.
Okay, yeah. Well, yeah, we have no good way to normalize that. But what we did is that we checked the injection site for each animal. For each animal, we inject same volume of virus, same batch of the virus. It's the only thing we can do. And in one slide, you can see that we overlay all the injection site of both animals in control each animal. And it looks very, very comparable. We did not see dramatic changes, but we have no good way to normalize this, how much virus you put in the brain and where you put we just get very careful histology. Also, yeah, it's very hard to believe that the variability in such surgery may keep reduction in EPSD amplitude so consistently. Thank you, but it's good, very good question.
Omar, you have another question?
Yeah, just comment on that. I mean, I think that is a big asterisk. And I think in all these kind of, not just yours, but any study comparing two cohorts of animals that have each individually been injected with channelrhodopsin, making a conclusive statement about their amplitudes being matched can be tricky. Obviously, your effect size is large, so that's fair. That everything should normalize in the wash, but it is, as you said, a very tricky thing to interpret. Next question is, you had the two groups of PV expressing neurons, the CC and the IN. And I know you classified them with your physiology index essentially, but how did you map those two classes onto the known two subtypes that are essentially only defined morphologically in the literature?
Okay, so we perform the cluster analysis first. We got two clusters, we call cluster 1, cluster 2. We have no idea what are they. Then we put their membrane properties together. We compare them. For example, the input resistance. One cluster obviously bigger than others. So then we just based on literature, which already reported that CC is have much lower input resistance compared to the interneuron. That's how we do that. In our paper was called putative CC. We have no direct reference.
Yeah, that's a tricky one. I mean, because so if input resistance varies a lot, even in your own graphs, like you have overlap in that. So for example, if you use the mean input resistance of one group to compare to the literature, that doesn't necessarily mean that every cell that you've clustered into that group is part of that group. And I have one final question, sorry. In the computational model from Bill Litton that you mentioned, you said that there was paradoxical increase in PT neuronal firing. What caused that in his model?
So from their work, I think they propose that increased firing rate of PT cells largely depend on the interlaminar excitation. Now, for example, the excitation input from layer 23 to layer 5, as well as the parameters, the input from PV cells. And I think this makes sense actually, based on our lessons from beta ganglion field, for example, in the STA, in GPE. After the stopping, those neurons also downregulate their intrinsic excitability, but after that, they are very sensitive to synaptic input. Like in ST area, this shows reduced intrinsic excitability. Their pathway become more effective in driving their firing in vivo. That's why in PBS and in animal models, people repetitively report the increase of ST activity in vivo. Maybe similar thing happened here in the cortical pyramidal. After they lose intrinsic ability, they are in vivo activity, which is mainly driven by the synaptic input.
Got it. Great, thank you.
Yeah, so if you thought about trying deep brain stimulation to see if you can also correct that thalamocortical change with chronic stimulation. Because DBS also has this sort of long-term effect when you turn it off. And that would be kind of cool if that were the mechanism that you change that synapse back and forth.
That's a great idea. Giving this very small ST mouse brain. That's
Yeah, I mean, you might not be able to do it in mice. You might have to, I mean, maybe in rats or
Yeah.
maybe epigenetically.
that's right.
Because that's definitely been done.
Yeah.
And then conversely, could you, right, if the mechanism of that change is that you get this sort of synchronized beta activity, could you induce that synaptic change just by stimulating without dopamine depleting? Like you chronically stimulated at 20 hertz.
I see, yeah.
Could you induce a similar change?
No, we haven't tried that, but we try to use using also genetics to silence activity, try to rescue them. That's right. That you know.
Does that work? Or we don't know yet?
No, we don't. Yeah, we did our next project to kind of small grant on that. We can manipulate from beta ganglia to thalamus to different subtype of cortical neurons, PV, which are interesting at different stages of the impact to the cortical circuit. Yeah, this is our next project.
So if the thalamus you think becomes mainly like inhibitory, like thalamic input to the PT, because it's going through these inhibitory PV neurons, which is so strengthened that the thalamus is essentially like inhibitory input to PV neurons. Do you think if you, in your experiments, put inhibitory opsin into the PV neurons and basically shut down that pathway, is there enough excitation left that you could stop the bursting, that you could kind of rescue some of that abnormal circuit behavior?
Yeah, great question. Yeah, we try to do that. Back in history, option or drag that in the PV field. According to what happened, we don't. Yeah, it's design, but we haven't done that. Great.
Questions? Okay, well, thanks again for a really fun talk. That was great.